Hello, everyone. My name is Zumra, and I'm excited to be here today as a new member of John Glass Lab. I joined the team 2 months ago, and I'm eager to share the project I will be working on. My focus will be on the development of whole genome transplantation techniques for non-mycoplasma bacteria. First, I would like to introduce you the current process of whole genome transplantation as shown in the figure at the bottom of this slide. We start with a synthetic genome that contains resistance marker in a bacterial or yeast cell. To boot up our synthetic genomes, we use a host or recipient cell, replacing the genome of the host with the synthetic one. This process has primarily been successful with Mycoplasma. I will walk you through the details of this method, highlighting key points that may be helping us for transition of whole genome transplantation to other bacterial species. Additionally, I will discuss potential candidate bacteria for this process. And finally, I will introduce a novel approach to visualizing whole genome transplantation using microfluidic devices. In 2010, JCVI SYN1 was announced in a science publication as the world's first cell with a chemically synthesized genome. This groundbreaking work involved synthesizing and installing a complete Mycoplasma mycoides subspecies Capri genome into a different species of Mycoplasma, resulting in a living synthetic bacterial cell. Following that, in 2016, the world saw the introduction of JCVI SYN3, the first near-minimal bacterium also equipped with a synthetic genome. It is important to note that up until now, whole genome transplantation has only been successfully performed in mycoplasmas. We believe that if whole genome transplantation can be adapted to facilitate the activation of synthetic genomes in a broader range of bacteria, it could significantly advance the field of synthetic cells. Currently, we possess the technology to boot up a cell with a complete synthetic genome in a single experiment. In contrast, constructing a genome piece by piece using current conjugation technology requires 35 to 40 steps to assemble an E. coli genome. This highlights the urgent need to understand whole genome transplantation and expand its applicability to other bacterial species, paving the way for the new era of synthetic cells. For preparing donor genomes, once we have grown the donor cells containing the genome, it is crucial to maintain their integrity throughout the process. To do this, we mix the cells with agarose gel to create agarose plugs. Next, we lyse the cells to eliminate them, leaving only the intact genome behind. Following this, we perform enzymatic digestion to prepare the donor genome for the transplantation process. To prepare our Mycoplasma capricolum recipient cells for transplantation, we treat them with calcium chloride and PEG. While calcium chloride ions mask the negative charges present both on the cell membrane and the donor synthetic genome, the PEG enhances the permeability of the cholesterol phospholipid bilayer of the cell membrane. Although the exact mechanism of this process are not fully understood, we proposed a naive starting model for the transplantation of tetracycline donor genomes into recipient cells based on the synthesia hypothesis, as you can see in the third image in this figure. Our transplanted genome has a tetracycline resistant gene. Only cells with this marker can grow in the presence of this drug. After cells have time to divide, we plate them on a agar containing tetracycline. Cells with a Mycoplasma capricolum genome will die because of this drug. The cells containing the synthetic genome will grow and produce colonies in about 3 days on the agar. In this section, I will discuss the identified issues associated with the mycoides mycoplasma system. Understanding these challenges is crucial as we may encounter similar problems when working with non-mycoplasma systems. We should focus on intraspecies rather than interspecies transplantation first. This work was conducted by Carol, a former member of Glass Lab, and represents her current work. Her findings indicate that the success rate of bacterial and yeast transplantation decreases significantly as the phylogenetic distance between the donor genomes and Mycoplasma capriculum recipient cells increases. We can see the importance of phylogenetic distance between donor and recipient cells. This study by Cannon and Gibson observed the surprising resilience of engineered 16S ribosomal RNA genes when subjected to addition of the genetic elements from phylogenetically distant bacteria. The researchers tested the function of these engineered genes through genome transplantation. For successful interspecies whole genome transplantation, ribosomal compatibility is really important. Given that there are no single ideal recipient cell that we can use for everything, we should definitely prioritize intraspecies transplantation to enhance our chances of success. The second issue, most of the bacterial cells have calcium-activated surface endonucleases. Trypsin can be used to removal of these nucleases before calcium addition. Many bacterial cells possess calcium-activated surface endonucleases, which Mycoplasma capriculum does not have. As we discussed earlier in the previous slide, calcium ions are needed for neutralizing the negative charges on the both cell membrane and the donor synthetic genome. However, these nucleases can degrade donor genomes, preventing successful transplantation. In this analysis, we observed the gel results for the PAC19 plasmid with Mycoplasma capriculum, Mycoplasma genitalium, a coliplasma Haemophilus species after trypsin treatment followed by calcium intubation. The simple gel assay reveals that the plasmid is significantly degraded after calcium treatment alone. In contrast, the combination of trypsin and calcium treatment demonstrates successful removal of surface endonucleases from the recipient cells, allowing for the preservation of the donor genome. To overcome calcium-activated endonuclease issue, trypsin can be employed prior to calcium treatment to help protect the donor genomes from degradation. Rubidium chloride has been effectively used to neutralize charges during the transformation of competent cells. Therefore, it presents a viable alternative to calcium for this purpose. Exploring rubidium chloride as a substitute would enhance the efficiency of the whole genome transplantation process. And the third issue, unlike nearly all of the other bacteria, our Mycoplasma capriculum recipient cells do not engage in homologous recombination. We can use antibiotic-resistant markers to select for the transplanted cells, as we have been discussing in Mycoplasma capriculum. However, this approach can result in false positives through homologous recombination for other bacteria. Therefore, it is essential to inactivate the recipient cell genome prior to transplantation to ensure accurate results. We attempted an intraspecies transplantation using Mycoplasma genitalium. In our initial efforts, employing a single selection marker led to homologous recombination within the recipient cells. In order to avoid this issue, we designed the donor genome to include two antibiotic resistance, as you can see in the picture. Their resistance markers located far apart from each other. The transplantation reaction was plated on nutrient agar containing both tetracycline and kanamycin. After 6 weeks, we observed 2 colonies. However, sequence analysis revealed that these were false positives, resulting from the combination of a large segment of the donor genome. specifically the region containing the antibiotic-resistant markers, into the recipient cell genome. Mitomycin C is a chemotherapy drug and known with its DNA cross-linking properties. It specifically targets a guanine nucleoside within the sequence of 5' CpG to 3'. Mitomycin C has cell-killing activity with its cytotoxic effects primarily during the G1 phase of the cell cycle and continues through the early stages of DNA synthesis. When Mycoplasma capricorn is incubated with mitomycin C, the majority of the cells die. However, we can still recover transplanted cells. This approach shows promise as a strategy to overcome the challenges of homologous recombination in whole genome transplantation for other bacterial species. So overall, we are motivated to expand whole genome transplantation from mycoplasmas to other bacterial species. With our current knowledge, addressing these identified issues represents the first steps toward creating new synthetic cells. We plan to start with intraspecies transplantation, iterating on trypsin treatment and mitomycin C for disabling the recipient genome. Additionally, we will employ spheroblast formation to degrade the bacterial cell wall, another feature that mycoplasmas does not have. So first, we are going to be plan to use a model gram-positive and model gram-negative bacteria. This approach will allow us to systematically explore the potential of whole genome transplantation across different bacterial types. Streptococcus thermophilus is a widely used as starter culture in the dairy industry. Its buffer is relatively cheap as it closely resembles milk. This bacterium has a small genome size, approximately 1.8 megabase pair. This table shows us the properties of the potential recipient and donor genomes of two strains. Notably, the potential recipient strain, LMD9, doesn't have surface-associated endonucleases. We will begin by optimizing transformation processes before advancing into whole genome transplantation. E. coli is a well-known and extensively studied bacteria that plays a significant role in gut health as a probiotic. Given recent successes in expressing larger genomes in yeast, we can now focus on a larger genome size, such as 3 megabase pairs reduce genome E. coli. While expressing such a large genome in yeast has never been achieved, accomplishing this would mark a new era in synthetic biology. Therefore, we will work E. coli as a candidate strain for whole genome transplantation. As you have seen, the steps involved in the whole genome transplantation procedure, they are tedious, and successful transplants are highly dependent on the operators. This highlights the urge of automated systems for this process. To better understand the mechanism underlying whole genome transplantation, we can leverage microfluidics technology to visualize whole genome transplantation process. This approach may provide us valuable insights and streamline for future experiments. Microfluidic devices for whole genome transplantation can be helpful for fully automated system. Automated pipetting reduces the damage of these large genomes while we are pipetting. Whole genome transplantation can be optimized for better results, and visualizing of a single cell transplantation can help us to understand real-time observation and analysis of individual cell behaviors. Mechanisms of whole genome transplantation can help us to understand the insights into the underlying processes, helps for further advancements. On the right, you can see an ideal microfluidic device designed for whole genome transplantation. This device begins with the loading of donor genome containing cells, followed by lysing them, and then the addition of recipient cells and treatment with calcium and PEG in order to successfully obtain transplanted cells. In practice, achieving whole genome transplantation through microfluidics may involve multiple devices. Therefore, our initial focus will be optimizing each step individually before integrating several steps into a system. Here's the first generation microfluidic device for whole genome transplantation. This is the initial attempt at a microfluidic chamber by James and Elizabeth. This chamber contains donor Mycoplasma mycoides genomes and Mycoplasma capricolum recipient cells, along with the addition of calcium and PEG. The results indicate that the synthesia hypothesis may be valid, providing a promising foundation for further exploration of the whole genome transplantation mechanisms using microfluidic devices. Now, I would like to thank John for his invaluable mentorship so far in this journey. I'm also grateful to my colleagues in the GLAS and Suzuki labs for their support. A special thanks to Elizabeth, Jane, Kate, and James for their contributions and encouragement during my studies. This is just the beginning, and I truly appreciate your assistance as I take my first steps in this exciting field. And thank you. And I'm happy to take questions.
Very nice, Zumra. Questions for her? Zumra, what's any thoughts about where you plan to go first? I know that you have now become a master at genome transplantation, and we talk about this a little bit. Which of the two alternative species do you want to take a crack at first, and why?
Okay, most probably I will go with the strep strain, because that one doesn't, like one of the strain, at least we know that it doesn't have the endonuclease, like the surface endonucleases. So I'm hoping that we can overcome the transformation of that one first. And also it has a smaller genome size. And we are going to be understanding new challenges, and we are going to be overcoming new challenges. And then we can switch the E. coli. That's what I am thinking of.